Hi, I'm Jan Pak from the Naoki Watanabe lab at Kyoto University. If you had to paint a face on the surface of a small coin, what kind of brush would you use? I'm interested in cellular dynamics, and of course we can look at these dynamics at several different layers, for example DNA or mRNA, but ideally we want to see what proteins do, right? Now, in traditional approaches, cells could be fixed and then stained with antibodies, or the target protein is fused to a fluorescent protein. Now, in this second approach, usually what is being done is that this target fusion construct is included in a vector which has very high expression levels of this construct. Now, this allows us to observe bulk and saturated signals in the cell, which can give you clues on the subcellular localization of the protein. But I'm more interested in the moment-to-moment actions of single proteins as they move throughout the cell. This is why I use a construct which has a very weak promoter that results in very low expressions of this target vector, of this target construct. And using this, I can actually track single molecules in living cells and see what it looks like in real time. In my project, the insulin receptor, which is crucial for metabolism and diseases such as diabetes, I want to know how the insulin receptor reacts to stimulation or inhibition. And to do this, I take downstream proteins of this insulin axis and fuse it to fluorescent proteins and then observe what happens under the microscope. And with this low expression level, I can actually track single speckles which represent a single protein and quantify and judge the reaction of the cell to either stimulation or inhibition. So this approach can improve drug research, and it can uncover hidden protein dynamics and actually elucidate protein behavior which we haven't seen before. So if you had to paint a face on the surface of a small coin, why not use the smallest brush that you have?